Good afternoon. My name is Tracy Bale. I'm from the University of Maryland School of Medicine here in Baltimore. Today, I would like to talk to you about some of our recent work focusing on trying to identify mechanisms and biomarkers using extracellular vesicles in circulation and as released from specific cells as hallmarks or biomarkers of stress signals, and identifying them in an approach that goes from animal models to humans and back. So first, let's discuss what are extracellular vesicles. So in order to first discuss the translation and reverse translationability of a biomarker, we first have to understand what this biomarker is and its mechanistic approach as to how it's derived. So extracellular vesicles are lipid bilayer particles that are cell-derived. They travel in circulation from most tissues in all mammals. Their release from all tissues and traveling in circulation has some order of specificity to it, largely dependent upon these proteins in the lipid bilayer of the extracellular vesicles. And extracellular vesicles, as we'll talk about today, come in many different flavors. And this is dependent upon the intracellular stores that they're released from and the contents of the vesicles and how they travel in circulation. They're largely thought to be important for intracellular and intertissue communication. Most of the work on vesicles has been done by the cancer field, where extracellular vesicles have been used as markers of cancer presence and in therapeutic treatment. So my lab is particularly interested in understanding stress and its effects on the brain, largely through development. How stress itself and its experiences preconception, during pregnancy, and during other key components of the lifespan can influence how the brain develops, how it matures, and how it functions and responds to its environment. We know that living in Baltimore City, that we are a majority Black city, and so the mental health of African Americans in this city, especially in the current environment, is critically important for us to understand. So 20% of African Americans adults are more likely to report psychological distress and mental health disorders than white adults. And we know in the current environment that these stress experiences and trauma, especially in the interurban environment, can be long-lasting and intergenerational. One of the biggest influences of stress, especially that of discrimination on relevant outcomes, is understanding the maternal death rate, where an African American woman with an advanced education remains in this country still 4 times more likely to die during pregnancy or the first year postpartum, and her offspring, so her child as well, than a Caucasian woman with only an 8th grade education. So understanding the influences of the environment on such outcomes as mental health and infant and maternal mortality are critically important for understanding the mechanisms of stress. The long-lasting effects and the compounding of these, the psychological toll taken by discrimination, stress, lack of resources, et cetera, in the environment is known as weathering. And this term has been coined by Arlene Geronimus years ago and has done a tremendous amount of work in this area. And trying to understand how the stresses that impact people of marginalized populations, especially experiencing significant discrimination, are chronic and repeated through the whole life course, meaning they can be compounded and produce effects on that generation as well as future generations. My lab is particularly interested in trying to define the biological mechanisms that contributed to those increased rates of affective disorders and as well as effects on mom and offspring. So one of the things we've been studying for well over a decade now is effects of stress in pregnancy and the outcomes in animal models and in human studies. We've now also added to this the effects of preconception stress. So mom's life experiences prior to conception, how they compound and can give rise to risk for both mom and baby. Some of that preconception stress can be defined as adverse childhood events. So those that have been traumatic events that have been experienced prior to adulthood. So in childhood, when those experiences occur and the types of those experiences have been shown to produce dramatic risk for many outcomes. There's a tremendous amount of this work that's actually been done at Columbia in this area. And so taking from a lot of those human studies, we've also been asking questions in our animal models to try and define, as it is, stress, compounding effects of stress, and when those effects of stress leave biological markers that we can further follow. So we know from the Kaiser Permanente study that assessed adverse childhood events that they assess them as being that have experienced 0, 1, 2, 3, or 4 or more, typically whereby the presence of 4 or more ACEs prior to adulthood was found to be a significant risk for many diseases across the lifespan, including many mental health disorders, as shown here, related to addiction, depression, diabetes, obesity, suicide, etc. So trying to understand the environment within which we're studying is critically important to understanding the risk factors. So if we take pregnancy as an example, and we look at pregnant women across the United States of America, and we assess current pregnant women for their ACEs that they experienced in childhood, you can see on average, here's shown in yellow at that 4 or more point of ACE experience, that across the United States, for 4 or more ACEs in currently pregnant women, it's about 12.5%. And actually, surprisingly, 36% that have had 0. But if you look in Baltimore City with women who would present at the University of Maryland Hospital current pregnancy and ask about their ACEs, you can see that the numbers are dramatically different. In this urban environment where there is a noted trauma and health disparities, that these women report as 4 or more at 61% of the population versus the 0 population, which here is shown as only 7%. So dramatic differences in the adversity and experiences here, which we would mark as preconception events, that no doubt will influence the course of pregnancy and the outcomes for both mom and baby. But how do we begin to understand the biological aspects of this? So we have a lot of knowledge now on the associations and understanding the risks, but trying to really get at the underlying biology and where we might intervene, I think, is critical. So my lab has taken much of what we know from many individuals studying these effects throughout the course of the lifespan, but also creating, looking at animal models where we can pinpoint specific biological signals that we may then be able to take back into humans and ask what the translationability is. So this includes for both dad and for mom in the preconception environment. But how do we really get at defining mouse models of stress? Certainly we cannot model discrimination. We cannot model the interurban environment per se as it affects of discrimination, stress, and the trauma that is experienced by many individuals. But we can assess the specific facts or the factors involved in stress experiences themselves. And one of the ways that we can do this is looking at the intersection of things like maternal stress at specific developmental points and looking at the cellular processes involved in responding to that stress. And we know that these cellular processes that might be involved are uniform, meaning that they are translatable, they occur in all mammals. So for instance, when post-conception or preconception influences on the conception event and early development, you can look at the level of the blastocyst shown here in purple, where the inner cell mass that will give rise to the developing embryo is shown in green, but these dark purple cells on the outside of the blastocyst here are the trophectoderm cells. And those cells following implantation in the endometrium of mom's uterus will then give rise to the developing placenta. If we can begin to understand both sperm and egg and the biological signals important at conception, and then follow the cells following that and begin to appreciate where preconception stress for mom or dad or stress in pregnancy begin to show differences both in the rate of the development of the embryo, right? Because the faster you develop, the more fine tuning that might be missed. So speeding up or slowing down the rate of development is key here. And you can follow that largely by looking at the transcriptome as it over the course of development. So over the last decade and a half, the lab has been dissecting the molecular mechanisms involved in how mom's intrauterine environment. So shown here as the F0, this is a rodent. So shown here is the F0 experience. So this could be maternal stress, it could be maternal diet, it could be maternal immune activation. Again, as many studies that have been done at Columbia as well. And looking at these effects of the intrauterine environment and how it gives rise to changes and the intersection at the placental development and those transplacental signals that give rise to the development and developmental rate of the somatic cells for the F1 generation here, as well as the germ cells that will give rise to the F2. And thinking also that the differences in the intrauterine environment may be dependent upon the sex of the developing fetus as well as mom's prior experiences and those currently during pregnancy. So in the last decade and a half, in the mouse model that we have been working on looking at maternal stress, we have developed a course of chronic variable stress experiences that mom has very early in pregnancy. So for the mouse, it's the first 7 days, which is pretty much the equivalent of the first trimester and the processes that are happening in a human pregnancy. And with these offspring, what we've found is that the male, but not the female offspring, remembering that mice have a litter, they're all growing and developing in that same intrauterine environment. But this phenotype of mom's stress was only passed on and affected her male offspring, but not the female offspring, as is often the case with neurodevelopmental disorders, that the males are at much higher risk for intrauterine environmental challenges. In our offspring, we see the changes in their behavioral stress response as well as their physiological HPA stress response. It's much more heightened and stress sensitive, again in the males, but not the female offspring. We see long-lasting changes in their central stress regulatory genes, cognitive changes in the way that they perform cognitive tasks. And we see this neuroendocrine phenotype in these males, whereby they're born normal for weight and growth, but once they hit about pubertal age, they sort of drop in their trajectory and they end up about 15% smaller than offspring who did not experience stress in utero. So all of this work has been published over the last decade and a half. I just want to focus in on one of the genes that we've identified as being a key node point for translating mom's stress experience at the level of the placenta, giving rise to these outcomes in the male but not female offspring. So through a transcriptomic approach years ago, we identified sex-specific genes in the placenta. One of these genes seemed a great target to follow up on. That is the gene I'm showing you here, O-glycosyltransferase, or OGT, which is an enzyme that is X-linked. And one of the reasons we followed up on this gene was because it's regulated by maternal glucose. So in the placenta, the energy availability by mom dictates and determines a lot about the regulation and processes of this gene. As it is an enzyme, OGT places what's called an O-glycanalylation mark, a sugar mark, on serine-threonine residues of hundreds of proteins. It oftentimes competes with phosphorylation events on those same proteins, whereby phosphorylation would be the gas of a given protein or the go for its activity. O-glycosylation or O-glycanalylation of that same mark is usually the break. So phosphorylation and O-glycanalylation tend to compete for activity of given proteins. And again, hundreds of proteins are regulated by O-glycanalylation. So it's known to be very important for processes involved in embryogenesis, really important for metabolism, immunity, and cardiovascular effects. But today I want to tell you at the level of the cell, especially within the level at the level of the placenta, it's epigenetic regulation. The reason for that is because as an enzyme, it's poised to regulate rapidly how a given cell, in this case, a trophoblast cell of the placenta, responds to the maternal environment, making it critically important to understand mechanistically. We know that OGT is X-linked. It seems in the placenta to escape X inactivation. So its levels are twice as high in a female cell, a female placenta, compared to a male placenta. And as we know, as I showed you a minute ago, that the trophectoderm, which gives rise to the placenta trophoblast cells, is the same sex as the developing fetus. And we know that OGT, being X-linked, is also positioned very close to the long non-coding RNA exist. And recent studies have actually demonstrated an interaction between O-glycanalylation and exist function, suggesting another route by which sex differences can rapidly respond to the changing environment, giving rise to different male and female outcomes in the environment. Okay, so this is some of the biochemistry from years ago that we identified. Not only is the RNA level poised based on X inactivation, escaping X inactivation, but as are the protein levels shown here. So it's twice as high, the protein, as in the female placenta compared to the male. This is true for mouse and for human. This is showing you that O-glycanalylation mark from total protein levels in a female versus a male placenta, showing you again that the protein levels determine the level of O-glycanalylated total protein in a placenta, again, twice as high. And what this means likely is that there may be some threshold by showing you here female placentas versus male placentas from the same intrauterine environment, either from a control here or from an early prenatal stress intrauterine environment, that while stress affects OGT in both males and females, that perhaps what is happening is that it's dropping below as an enzyme, that this enzyme level is dropping below some threshold of vulnerability, meaning that the level the levels of oglyconacylated proteins that are important for transplacental signals and placental function have now dropped to a point in these animals that you're seeing altered signals being delivered to the developing fetus. Okay, so think way down in now at the level of the trophoblast cell in the placenta. What does OGT do that might be important for understanding cellular function? One of the main things that oglyconacolation does is it stabilizes, especially as related to transcriptional responses, is that it stabilizes a methyltransferase, EZH2. So EZH2 is stabilized by oglyconacolation. When it is around more, and there's more of EZH2, you get more methylation of histone 3 lysine 27. So we know that histone 3 lysine 27, when it is more methylated, meaning H3K27 trimethylated, is a standard inhibitory transcriptional histone mark. So it is a transcriptional repressive mark. I don't have time to go in today to the UTX and UTY regulation of demethylation of this mark as well. That's another time. But our hypothesis was initially that if OGT is regulating and stabilizing EZH2, if you have more OGT, then you should ultimately have more H3K27 trimethylation. And in fact, that is what Bridget Nugent, a postdoc at the time, found exactly. So if you compare, again, a female placenta to a male placental from the same intrauterine environment, you see a lot more of this mark in the female than the male. Interestingly, this mark is also required as part of that process of exon activation. So it's fitting that you would see more of this repressive mark in a female tissue than in a male tissue. Okay, so as an example of why this might be important for placental function, I'm showing you some studies that we did here looking at sex differences and transcriptional regulation in the placenta using ribotag mice. So we're looking at the active translatome from the placenta, only from those cells that are from trophoblast lineage, comparing transcriptional differences at one time point. This is at embryonic day 18.5, so a full day before birth in a mouse, right? This is right before birth, comparing the transcriptional differences between a female placenta and a male placenta from the same intrauterine environment, right? Across many, many different pregnancies. Okay, so on the y-axis is log two-fold change for increased and decreased levels or transcriptional differences between males and females. And then the level of gene expression here, the normalized counts is on the x-axis, meaning highly expressed genes are out here, which obviously there's fewer of them. Lower expressed genes, because this is RNA sequencing, we detect lots of those, is here. And what you're seeing with the red dot is indicating a robust, statistically significant difference in all genes shown here that are expressed between males and females. Okay, so lots and lots of red dots, lots of differences between males and females across high and low expressed genes. Okay, so if we play around with the level of OGT, meaning if we use transgenic mice to eliminate OGT from one of the X chromosomes in a female placenta cell, so that her level is the same as a male, right? So a male is XY, the female is XX. So we're reducing her OGT levels only in the placenta, nowhere else. And now saying if we eliminate the differences in OGT between male and female placentas, now what does transcription look like? And you can see that all those differences are almost all those differences are eliminated, suggesting again that this OGT enzyme plays a really important role, possibly through the stabilization of EZH2 and regulation of H3K27 trimethylation. And in fact, when Bridget has subsequently looked at levels of H3K27 methylation, they correspond, of course, with the levels of OGT. And in fact, these red dots represent genes that are clearly not involved with OGT regulation, but however, are themselves either X or Y linked. So this is a unique way of assessing potential sex differences that may guide placental responses to the maternal environment and thereby changing transplacental signals important for fetal development, especially that of fetal brain development. Okay, so what about the brain? If we then take these same animals that we took the placenta from here on the right, and we now ask about the brain. Okay, so this is using RNA sequencing from the E18.5, again, the day before birth in a mouse. So on the left is showing you the brains from the normal female placentas. So we did not alter OGT. And on the right is from the normal male placentas. We did not alter OGT in the placenta. Okay. So what you see here at embryonic day 18.5 in the hypothalamus of these animals Are there known and predicted sex differences? We know that hypothalamic development contains a lot of differences in the rate and maturation of a lot of the nuclei within the hypothalamus for males and females. So this is not a surprise. But what is a surprise is if you lower the OGT to be similar between a male and a female in the placenta, and then ask what happens to the female's brain. And in fact, what happens is that she looks a lot more like that of a male brain at this developmental time point than a female brain. And what that likely means is that those transplacental signals, guided and regulated by levels of OGT in the placenta, are giving rise to transplacental cues to the developing fetus that are altering potentially the rate of how this brain is developing. And that may be important for our understanding, both for trying to determine what those transplacental signals are, but also what is important for fine-tuning the brain and how that rate of development matters. So the lab is currently trying to mechanistically define and determine what those transplacental signals might be and how are they different. So if you think about what the placenta is doing, it's intersecting between mom's maternal milieu and environment and providing its own, because the placenta is a very highly endocrine tissue itself. So it's responding to mom's environment, it's processing, making a lot of key proteins, some steroid hormones, all kinds of metabolic factors required for energy and development of the developing fetus, especially the brain. Another thing that the placenta does is it makes a lot of extracellular vesicles. And in fact, across all mammals in circulation, the highest level you'll ever find of extracellular vesicles traveling in circulation is during pregnancy, because the placenta makes so many vesicles. And as I told you earlier, the cancer field has done a tremendous amount of work looking at extracellular vesicles and their signaling, a majority of which has been found around signaling to the immune system. So that makes sense that during pregnancy, you would want to be signaling to the immune system of mom, allowing for the development of the fetus. Okay, so back to extracellular vesicles. As I said, the extracellular vesicles come in many different flavors. There are very small types of extracellular vesicles, which are called exosomes. So exosomes are a type of extracellular vesicle. There's larger extracellular vesicles known as microvesicles, and there's a variety in between. They differ not only in their size and their composition, but also from the intracellular compartment of any given cell from which they are processed, the cargo is loaded, and how they're released from the cell. So trying to understand, so Joel Sadowski has done a tremendous amount of work in this area, dissecting the factors of placental extracellular vesicle secretion, how it signals to mom, how it might signal within the intrauterine compartment, signaling within the fetal compartment, as well as trying to understand within the placenta itself. So interestingly, just like endocrine factors can also be autocrine and paracrine, so can extracellular vesicles. Okay, so there are many different things that you can study again about an extracellular vesicle. If you're collecting them in circulation, because there's no way to know where they're coming from, you can look at many factors and try to sort out by many different ways where the changes might lie in a vesicle and the signaling it might be producing. So there's the proteomic approach of understanding the protein content. The protein content of this lipid bilayer of these particles can tell you somewhat of the tissues or the cells it might be coming from, as well as the tissue or cells that it's traveling to, because there is specificity for that. There's also the small non-coding microRNA or small non-coding RNA content or cargo of these vesicles, largely microRNAs, but other content as well. So you can also perform a transcriptomic approach on the content of these vesicles. Also of interest, as I just told you, that extracellular vesicles importantly have a difference in size, and that can tell you a lot about, again, the intracellular compartment and the tissues they're being released from. So using nanosite technology allows you to assess the size, the average size, and the heterogeneous composition of what you're collecting for extracellular vesicles, and can show you if whatever your parameter is, such as stress, is changing or shaping the overall size of the extracellular vesicle pool you have. Okay, so I just want to tell you one story that we've recently started on in understanding extracellular vesicles and maternal stress in pregnancy. So this is from our animal model. This is looking at, on the left, the transcriptomic approach of extracellular vesicles and our preliminary data here, and on the right, more astoundingly, the proteomic data. So first on the left, if you take extracellular vesicles from circulation from a mouse, on the far left are showing you 4 or 5, I guess there's 4 different animals here that were not pregnant. And showing you on the far right are the microRNAs that were statistically different between these groups. So you can see just by the color of this heat map, the biggest difference is the state of pregnancy itself. Whether you are a control pregnant animal or an early prenatally stressed pregnant animal, the biggest difference, no surprise, is the content of the EVs is related to pregnancy. So that supports the idea that extracellular vesicles in relation to placental release into mom's circulation promote a dramatic difference. But we did find that there is initially some evidence of some really important microRNA cargo here that are statistically different with maternal stress. Okay, but remember, these are also from circulation. Okay, so we don't know exactly what tissue they're coming from. But also of incredible interest was in comparing the effects within pregnancy of stress on mom and the vesicles in circulation, that we saw really big differences by a proteomics approach in proteins, the protein content of these vesicles, with a statistical reduction in the immune system and immune system processing of these protein content of the vesicles, and a huge increase in relationship to metabolic processes. So I think one of the things that we're now thinking about how to follow up on these studies is begin to look at the specificity of the placental cells and the cell types differentiation, for instance, and how they might give rise to differences in the EV cargo and what those might mean. But one of the things we want to know is, do the maternal circulating extracellular vesicles, because they're so teeny tiny, do they make it into the fetal compartment and have an effect on fetal development? Or are they limited to interactions with the placenta and in mom's compartment, the maternal compartment? So we were able to isolate extracellular vesicles from stressed and control moms during mid-gestation and fluorescently label. So we concentrated the EVs, fluorescently labeled them, and then we can inject them back into a normal pregnant female and ask, where do they go? So what you can see here, looking at the signal, that they're really only aligning with where the placenta is. And in fact, if you dissect out that intrauterine compartment here as the placenta and the fetus, we don't see any signal making it into that fetal compartment 24 hours after that injection has happened. So it's not to say that they can't get in. It's not to say that there isn't some level. We're just not detecting a robust level here. They're really interacting at the level of the trophoblast placental cells itself. But that too is important for understanding its interaction, both as Joel Sadowski has shown at the level of the placenta itself, but also in the maternal compartment as well. So back to understanding its effects at the human level and trying to understand the effects of discrimination stress or weathering, and that high increased risk as it might give rise to infant mortality and discrimination stress in pregnant women and their offspring. So one of the things that we've started doing is a collaboration with Tanya Jovanovic as part of the Grady Trauma Project. So Tanya is now at Wayne State developing the Detroit Trauma Project, but she followed up on managing the Grady Trauma Project at Emory after Carrie Ressler, who had started the Grady Trauma Project, moved to McLean. So Tanya was in our collaboration. They provided us with plasma blood samples. We recruited women asking the question about trauma experience. We wanted to know if there was a timing specificity. So this is really just very new data that we've done looking at extracellular vesicles in this population. So these are majority Black women in this population. We asked about interpersonal violence, specifically looking at sexual trauma. And we looked at it as no sexual trauma, or women who experienced it under the age of 14 during adolescence, so the ages of 14 to 17, or outside of childhood, they're in adulthood over the age of 17. And then we did a lot of physiological measures, which I don't have time to go into today. We also collected blood samples, and we looked at circulating cell-free mitochondrial DNA, cytokines, and extracellular vesicles by proteomics and small RNA sequencing. I'm not going to go through all of the data with you, but the point that I want to make in these studies is that what we're starting to see is some pretty profound differences in these biomarkers and their relationship with the stress, physiological, or trauma experience in these women. So first, if you look at now these women are adult, so they're in their mid to late 30s. This is showing you body weight for no sexual trauma versus overall sexual trauma, which we see a difference in increase in body weight. And then if you parse it out by the age of that sexual trauma experience, you see that this is largely driven by women who experienced sexual trauma before the age of 14. The same is true for hip circumference, for BMI, and for waist circumference as well. We see the same factors. That again, it is largely driven by this group of women who experienced that trauma prior to the age of 14. Also of interest, when you look at blood pressure in these same women, which fits with their body mass index and weight gain, that you can start to see that overall the high blood pressure here categorized for sexual trauma experience versus not is higher in these women. If you then parse it out again by age of experience, that hypertension stage 2, which is the green one here versus 1. So basically looking at movement into a very high risk for hypertension at a younger age, you can see by age of sexual trauma experience is higher here for women under 14 when they experienced it, then adolescents, and then lastly over 17, which again suggests that there is a biological mechanism or a biological underpinning for when the trauma experienced and its lasting effects on health outcomes. So also of interest, when Tanya's group performed an assessment of fear potentiated startle on these individuals, we see something different. That again, we see the factors being driven again in the women who experience sexual trauma. But again, this is a different group. Now it's the women who experience sexual trauma during adolescence are showing higher rates of fear potentiated startle here, no difference in baseline startle, but also if you're going to look at the skin conductance rate here, we're also seeing difference in this particular age group. So very different outcomes, more related to PTSD versus metabolic effects in the two different age groups of sexual trauma experience. Okay, so we then wanted to look at the biomarkers here. So we did several different things, which I don't have time to get into, including cell-free mitochondrial DNA levels, which are also higher in the women experiencing sexual trauma. But I want to quickly focus on the extracellular vesicles. So this is showing you a proteomics approach that Kathleen Morrison did as a postdoc in the lab, comparing no sexual trauma versus sexual trauma overall for all proteins detected in circulation in these EVs. You can see dramatic differences in these two populations. But what I want to draw your attention to is the huge differences we see across, so some similarities for sexual trauma experience here in these proteins, which are much more lowly expressed in these women compared to no sexual trauma, the pink at the bottom here, versus in this heat map, the top in this one specific group, the adolescent women who experience sexual trauma during adolescence, this blue group here. And what these proteins are is they're largely related to keratin and keratin-related proteins, which I'll come back to in a second. So in Katie's assessment, what she pulled out from this proteomics approach was, this is again, log twofold change by proteomics of these really, really important proteins. There's many more, but I'm just showing you these 4, profound reductions in the levels of annexins and galectins. And these proteins are known to be really important for determining how extracellular vesicles are loaded in the cell, how they're released. So it has a lot to do with the extracellular vesicle function. And it's specific to different types of tissues. So this can tell us a lot about which tissues we might be seeing a reduction in vesicular release from those tissues in circulation, what that might mean for signaling processes. But the really amazing difference that we found, as I said here with these keratin proteins, only in women who had experienced sexual trauma during adolescence and in no other group, we saw a huge enrichment. So if you see here, the log twofold change, these are not subtle differences in a large number of keratin associated proteins and cuticular type proteins. So these are hair and skin related proteins, skin cells called keratinocytes, which are known to be very important for release of extracellular vesicles into circulation. Only in this one group, an enrichment of those proteins. This suggests to us that trauma experienced during adolescence may be integrating signals at the level of the skin, specific types of cells at the skin, possibly keratinocytes related to hair production as well. And that this signal may be part of, as the skin is your largest organ, and is a key and important process along the stress axis, one we often forget. So along trauma and stress responses, this may suggest something very novel that we had never identified before in our stress discovery, and understanding stress programming as it relates to signals important for how trauma may be integrated in a given individual. Also a follow-up to this, we've just recently discovered that the majority of these keratin and keratin-related proteins belong to a gene cluster on the chromosome 17. So that's also of potential interest for an epigenetic regulatory mechanism. Okay, so quickly I want to jump to going back to understanding the preconception environment and how we model this in animals and may translate it into humans. So as you may know, of course, at fertilization, we have the egg for mom. So her preconception environment can alter what is present at conception in the egg. But we also have a lot of sperm coming from dad. And only one sperm may actually fertilize the egg, of course, but many, many sperm, up to millions of sperm, can show up at the egg and bring hosts of signals with them. So what is it about dad's preconception environment? We know a lot of history, both from the Dutch hunger winter and the Swedish famines, where there's a specific unique region called the Overkalix region in northern Sweden. We know that this is critically important. The studies being done there for understanding the processes of famine and its programming, especially in boys, for what they passed on to multiple generations for risk and resilience, longevity, health, diabetes, as well as mental health risks has been studied in these individuals. So we know that there's an association between dad's preconception environment giving rise to biological processes important for how his offspring grow in development and their risk for disease. In fact, the New York Times ran an article about 2 years ago, almost 2 years ago now, where they began looking at Civil War outcomes and tracing effects on offspring. I think this just highlights the fact that this has become much more accepted now, that it's not just mom's preconception environment, that dad's preconception environment is becoming a significant factor for outcomes as well. So almost a decade ago, we had begun developing a paternal stress model. This is just a summary of what this model examines here. This is taking male mice. And wanting to be translatable, we began studying the effects of stress in the dad's environment prior to pregnancy, prior to conception. The males are exposed to 4 weeks of chronic stress. They are bred immediately after the end of stress or 3 months later, and I'll come back to that. They produce male and female offspring. Doesn't matter what the sex is here of the offspring. They both show the same effect, which is a significantly hypo-responsive or less reactive stress axis. We've identified some of the markers in the sperm. I'll come back to that. And we see broad transcriptional changes in the developing brains of these offspring. So I just want to quickly show you what this looks like in their offspring. These are the male offspring of stressed dads. These are the female offspring of stressed dads. Showing you here work done by Jennifer Chan, who was a graduate student in the lab and now is a postdoc with Ian Mays at Sinai. Showing you here is an HPA stress response in a mouse. So we're measuring corticosterone levels on the y-axis here. This is an acute restraint to these animals when they are adults. And so what this is showing you is that if we stressed dad, chronic variable stress, or we did a chronic variable stress double model, is what we called it at the time, just to make sure we were maxing out dad's stress experience. We give him plenty of time off after the end of stress, and then we quickly breed him so he's not with mom for more than 3 days. And then we look at the developing offspring. So dad never interacts with his offspring. And this effect is long lasting. So he can produce it with multiple litters, multiple females. He can produce this effect on offspring even 3 months after the end of stress has occurred. And all this work is published. So this is just showing you that if we breed dad immediately at the end of his stress experience, he does not pass on an effect, no matter if it's a single stress that a single type of daily stress he experienced, or even the severe stress I showed you, that the offspring show no difference if dad is bred right at the end. So this is modeled here up at the top. F0 is the dad. He's been exposed to 4 weeks of stress. He is either given a week off and then bred, or he's given 3 months off and then bred. What's interesting, as I showed you before, is that if he's bred a week after, he doesn't pass on the effect at all. And in fact, it requires a time post-stress recovery to allow a new allostatic set point, we think, in the somatic cells of the reproductive tract. And what we see here is if we look at the sperm microRNA, so the small non-coding RNA content of dad's sperm, this is the mouse. If we compare a control, not stressed dads versus 1 week after stress is over, almost no differences in the overall microRNA content. However, if we look at three months after stress has ended, here's the controls on top, here are the stressed dads. We see major differences between these two groups in the microRNA content of his sperm. Now, sperm are transcriptionally inert, and we know from the many studies we've done in the lab that this process of stress experience does not occur during the course of spermatogenesis. We know that it occurs in the maturation process that occurs outside of the testes. It occurs in these tubules called the caput, or the head of the epididymis. As sperm are transcriptionally inert, that means that they cannot respond to the environment. They are not themselves Sorry, they are not themselves transcribing these microRNA. That's not possible in a sperm. So how is the microRNA content associated with sperm changing in response to the environment? So this is the model from Jennifer Chan's work, showing you again, this happens outside the course of spermatogenesis at the caput region in these tubules of the head of the caput. These tubules have epididymal epithelial cells that secrete all kinds of important growth factors and processes, including extracellular vesicles, into the tubule where the sperm are maturing. This process takes weeks to occur in all mammals. And in fact, these sperm are put into, due to the pH of the tubule, sort of a static state so they can receive all of these signals required for their maturation. Prior to this process, sperm are not mature, they cannot fertilize an egg, and they cannot swim. So this process is critical. So it is this point that we believe that dad's environment, through its effects on these somatic cells, these epididymal epithelial cells, where dad's experiences can promote long-term, long-lasting changes at the epigenetic level of somatic cells that alter the course of what is secreted into the tubule here and interacting with sperm that allows these lasting changes to occur. And in fact, it is the allostatic set point whereby when stress is over, that the epigenetic changes occur and the differences then are detected in the sperm that are delivered at fertilization. So to test that hypothesis, this brilliant graduate student developed what we call the stress in a dish model. Why she had to do it in a dish is because there is no possible way, and she tried, to collect extracellular vesicles from a mouse epididymis. The tubules are just too fine. You cannot get this specific population if you wanted to ask the question of what is the evidence that following stress, the content change you see in the sperm is coming from the vesicle. So instead she had to do this in a dish where she was able to culture these caput mouse epididymal epithelial cells, they're called DC2 cells, in a dish. And by treating those cells in a dish with glucocorticoids and looking at a dose response and a time response pattern similar to what she did in vivo, able to collect extracellular vesicles in the conditioned medium. This is just showing you all of her work that has been recently published. And in fact, looking at what she wanted to do as an expert pharmacology graduate student, this is showing you a rank geometric correlation here, showing you if you look at the in vivo sperm changes in microRNA and correlate them with the changes being increased or decreased with what she sees in the EVs in culture, she was able to assess the time course of stress treatment, so 3 days is what it required, and the time course post-stress end, so 3 changes of media and 8 days later, as being the point at which the stress level change of microRNA in the vesicles and culture matched best with that of the sperm in vivo. And that's showing you the EV microRNAs here, showing you with no treatment, just a baseline low level of glucocorticoids, and then the stress level over those 3 days, showing you dramatic differences in the microRNA that are very similar to what she saw in vivo in the sperm. And this is showing you really interesting differences in looking at the vesicles from the conditioned media and culture from these epididymal epithelial cells, showing you at the end of the stress treatment, there's very little difference looking at this orthogonal score here of the protein content, but 8 days later, now you're seeing dramatic differences as shown you here in this heat map, comparing the vehicle treated EVs versus the previously court treated. 8 days later, you see dramatic differences again in that protein content of those extracellular vesicles. What's also really interesting, because again, this is a pure cell population, so you can learn a lot from how EVs operate here. This is using nanocyte technology that uses particle tracking. And you can see the stress effects on these EVs is not just protein, but the size of the EVs is reduced. And this is really interesting because the glucocorticoids are producing a lasting effect on what's being secreted, right? So maybe driving it toward a more exosome type of vesicle versus a microvesicle. That's interesting. But this has been seen before in the literature with stress effects, reducing the size of vesicles. And it's also been shown in other species, in humans and in sheep. So it seems to be something about the glucocorticoid process, possibly that allostatic set point, that is shaping and changing the type of vesicle being released. Okay, so just to quickly tell you what this brilliant grad student did in her last study for this paper was she wanted to ask a causal question, right? We've done a lot of associations. We see overlap in her stress in the dish with the in vivo, but she really wanted to do the causal assessment. So she did her stress in the dish protocol. She collected her extracellular vesicles at the same time point I just showed you, 8 days after. So these are vesicles from vehicle treatment versus cord treatment. She then collected sperm from one dad. So the dad himself is a control mouse. She took the dad's sperm and divided it into 2 pools. She incubated that sperm with either the previously vehicle treated vesicles or the previously cord treated. So this is the only difference now is the vesicles, washed all of the sperm. She then collected eggs or oocytes from a female mouse that has been hormonally stimulated. She divided those eggs into 2 pools, performed intracytoplasmic sperm injection, a normal fertility method, whereby the sperm from this pool were injected into one pool of eggs, and the sperm from this incubation was injected into the second. Okay, so the sperm is from the same dad, the eggs are from the same mom, the only difference being the vesicles they were incubated with. So then she did 2 different things. One, she transferred a pool of these ICSI performed zygotes into the left or right uterine horn of the same female. Okay, so she did that experiment initially. And then at mid-gestation, she then took out those developing placenta and fetuses and did a transcriptomic assessment to ask if sperm from the same dad, eggs from the same mom, in the same intrauterine environment, do these vesicles make any difference? And then she also performed a second level of experiment where she did them in separate ones and let them be born normally and develop and asked about their stress reactivity. So this is the amazing data that she found as proof of causality, that when she performed this very complicated experiment and did a mid-gestation transcriptomic assessment, she found that in the brain and the placenta, there's dramatic differences here in synaptic signaling related transcriptomic approach and neurotransmitters. So this is using RNA sequencing mid-gestation, that the brain, likely the rate of brain development, has been altered by incubation of sperm with those extracellular vesicles from culture, showing here. And then when you let the animals grow up, the male and female offspring have a hypo-responsive HPA axis, just like she saw in vivo. So again, demonstrating, going back and forth between in vivo and in vitro results, that these differences in extracellular vesicle content seem to make a big difference for the fetal brain development and how those animals then react to their environment later in life. So I just want to end with how we take this then translationally. So in a collaboration with Neil Epperson, these studies actually began when Neil and I were both at the University of Pennsylvania and took several years to complete because they were very complicated. We basically recruited Penn undergrads and medical students. Because what we wanted to know, how these studies started was, we can assess in mice the content, the small non-coding RNA content of sperm in mice. But what we wanted to know in humans, what is the variability across a given subject? So within subject and between subject over the course of 6 months. So if you got them to come in and do a donation every month over the course of 6 months, and of course we paid them very handsomely to do these studies. And then Neil, being the psychiatrist, her group assessed them for perceived stress for each month. They did an initial assessment, of course, of all of their adverse childhood events, assessment, etc. And then we then collected the sperm samples. We ran small non-coding RNA sequencing, all of the different types of small non-coding RNA. And then over the course of the last year here at Maryland with the IGS, the Institute for Genome Sciences, we developed a very complex bioinformatic modeling that took into account their perceived stress score for the preceding month, and then modeled that as predictive for changes in the small non-coding RNA content for the T+1, T+2, T+3, etc. over the course of stress. The question being, and these studies have just come out in scientific reports, can we identify within subject and then between subject any small non-coding RNA, whether it be microRNA, tRNA fragments, or piRNAs that are consistently dynamically responding to the environment? Once we develop this framework, You would then be able to ask questions for any other interests that you had. For instance, dads who have children with autism, dads who have children with schizophrenia, dads who have come home from military service and been experiencing PTSD, etc. But it's very difficult right now to ask those questions because there is no normal framework to build upon. So that was our goal. So this is just a snippet of the data to show you here. This is looking at the top A here is showing you microRNA. The middle is showing you tRNA fragments, and the bottom is showing you piRNA. This is just showing you representation from 5 of our subjects. Subject number is as here on the top, subject 13, subject 2, subject 22, etc. So we had about 20 subjects complete this assessment. What I'm showing you here, which you have to, if you look very close, you can see the dark blue are the small non-coding RNA. So in this example here, microRNA, that are statistically significantly different in response to the environment or highly expressed across subjects, across all subjects here, showing you their expression pattern. So if they're deemed dynamic in at least a majority of our subjects, I'm showing you the same ones across all subjects. If you look closely, you can see all of the microRNAs, the light blue, so it's not a light blue background, those are all different microRNAs. There's hundreds of microRNA in sperm. Okay, so what you can start to see here is that sometimes there are patterns of dynamic expression. So in a given month where something is very, very high, the next month it might be low, etc. There's fewer of those for the tRNA fragments. But what is really interesting, and I hope you can see here, is that we've noted a pattern of expression across almost all subjects for the piRNAs present in sperm, whereby there seems to be an every other month cycle of how these piRNAs, a large number of them, are being expressed. And that is super interesting because piRNAs previously were thought to only be important for the course of spermatogenesis, but this would suggest that there is some factor here with the piRNAs in mature sperm that may be some signal related to fertility or the environment. We don't know, but it's super interesting and something to follow up on. Okay, so when we actually assess using our robust calculation and bioinformatic approach between subject and within subject, we actually were able to identify a total number within each small non-coding RNA group, a total number that were different. Those that fit our model of being dynamic, meaning they had to give rise to going from a low expression to a high expression within a period of time across months. And then from those, correlate with an individual's perceived stress in the prior months, we were able to identify 5 specific microRNA that fit that pattern across all of our subjects for microRNA and tRNA fragments. However, no piRNAs fit that model. So that is really exciting because it suggests that there may be common processes across subjects, so across humans, that are responsive to this, in this case, stress in the environment or resolution of stress in the environment, as it may be in this case, that could produce differences in offspring at fertilization. Because many of these, just as 2 examples, LET7F and MIR181A are already known in the world of fertility and reproductive outcomes to be really important. So these may be stress signals we've identified in sperm that lower levels or higher levels might occur in response to stress in the environment, giving rise to signals that determine the rate of fetal development. And so now our studies are ongoing where we can take those specific identified small non-coding RNAs, inject them in mice zygotes, and then ask about the course of development. Those studies are ongoing. So just to finish up here, I hope that I have demonstrated to you that extracellular vesicles are an incredibly important stress signal or biomarker, as it may be. We've gone everywhere from looking at them in very, very specific cell types, such as epididymal epithelial cells, as they give rise to important stress signals, again, related to the course of fetal development, probably the rate, but also in relationship to discrimination stress and how factors related to interpersonal violence trauma experience may also give rise to important biomarkers that may signal the immune system, or for that matter, stress reactivity involving the skin, studies we're following up on. So with that, in summary, I've already summarized a lot of this, but the OGT signals we think are key for a lot of the extracellular vesicle secretion from the placenta, and those signals we're currently studying, that there are timing specific trauma outcomes related to when trauma in development, especially in childhood, produces lasting effects and at what tissue level. And that EVs and a very specific outcome, looking at vesicles produced from epididymal epithelial cells interacting with a germ cell, may play a really important role in guiding the rates of embryo development that impact ultimately the brain. So I hope I've convinced you that EVs may serve as a really important stress signal as well as an important biomarker in humans. So this work obviously is done by amazing lab members. Bridget Nugent was a postdoc who did all of the work on the OGT and the placenta studies. Katie Morrison has followed up on that and done the studies with the Grady Trauma Project in collaboration with Tanya Jovanovic and Annie Stenson and Sierra Carter, who are now at Georgia State and Emory. The studies done in the lab related to the sperm studies were started by many members of the lab, but the main work that I showed you today was done by Jennifer Chan, a graduate student at that time in the lab and now a postdoc with Ian Mays. And Christopher Morgan is a postdoc in the lab working on all of the human studies as we launch into our next cohort of men being recruited now that Neil Epperson is chair of psychiatry at the University of Colorado in Denver. I want to thank our other collaborators who helped us with a lot of this work, including IGS members here at Maryland, our collaborators at USC as part of the Grady Trauma Project, and Epigenetics Institute at Penn. Please follow us on Twitter, as we're always engaging in the Baltimore community and working to understand the effects of trauma in the Baltimore community and its effects on the brain, and all of our funding sources from NIMH, NICHD, and NIEHS. And I am happy to take any questions. Thank you.